Lion's mane mushroom, Hericium Erinaceus, has gained attention for its potential neurological benefits. Within the realm of neuroscience, lion's mane is believed to offer advantages due to its compounds, particularly the hericinones and the erinacines, which have been shown promising effects in preclinical studies. The neurological benefits of lion's mane primarily revolve around its potential to support nerve growth factor, NGF, production and related pathways. NGF is a protein crucial for the growth and maintenance and survival of nerve cells or neurons in the brain and peripheral nervous system. Lion's mane's active compounds, especially erinacines, have been shown in some studies to promote NGF synthesis. This is crucial because NGF helps in the formation and maintenance of neurons, thereby potentially aiding in nerve regeneration and repair. Such effects could be particularly beneficial in conditions where nerve damage occurs, like neurodegenerative disease or after injuries. Understanding the pathophysiology behind lion's mane's neurological impact involves delving into the mechanisms of NGF production and its role in neuronal health. While lion's mane's extract is consumed, its bioactive compounds can cross the blood-brain barrier and reach the brain. Once there, they have been observed to stimulate certain cells, leading to the increase in NGF synthesis. NGF operates by binding to specific receptors on neurons, initiating a cascade of signaling pathways. These pathways ultimately regulate neuronal survival, growth, and differentiation. By enhancing NGF levels, lion's mane potentially supports these processes, aiding in neuronal maintenance and even potentially facilitating the regeneration of damaged nerves. Moreover, lion's mane's potential benefits extend beyond NGF modulation. It possesses antioxidant and anti-inflammatory properties, which are valuable for neurological health. Oxidative stress and inflammation can damage neurons and contribute to various neurodegenerative conditions. Lion's mane compounds, by reducing oxidative stress and inflammation, may help protect neurons from damage and slow down the progression of certain neurological disorders. Research on lion's mane's neurological effects is still evolving. While promising, it's essential to note that most studies have been conducted on animals or in vitro. Clinical trials on humans are ongoing but limited. Therefore, while preliminary evidence is exciting, more robust clinical studies are needed to fully comprehend the mushroom's effects on neurological health. The neurological benefits of lion's mane are primarily linked to its ability to potentially enhance NGF production, support nerve growth and maintenance, and provide neuroprotective effects through its antioxidant and anti-inflammatory properties. Understanding the detailed pathophysiology mechanisms involving sheds light on its potential therapeutic applications in neurological disorders, but further research is required to validate and fully comprehend its neurological benefits to humans. The specific cells that lion's mane compounds are believed to stimulate for the increase in nerve growth factor, NGF synthesis, are astrocytes. Astrocytes are a type of glial cell found in the central nervous system, CNS, and play crucial roles in supporting neuronal factor function and health. Studies suggest that certain compounds present in lion's mane, such as erinacines, may stimulate these astrocytes. When activated, astrocytes can increase the production and release of NGF. This released NGF can bind to receptors on neurons, initiating a signaling pathways that support neuronal survival, growth, and differentiation. This stimulation of astrocytes by lion's mane extracts is believed to be one of the key mechanisms through which NGF levels are elevated, potentially contributing to the mushroom's neurological benefits. While NGF, nerve growth factor, primarily influences the growth, survival, maintenance of nerve cells, neurons, it doesn't directly participate in myelination. Myelination is the process in which neurons are insulated with a myelin sheath, a fatty substance that speeds up transmission of nerve impulses. However, NGF does indirectly influence myelination. It plays a role in the development and survival of the cells that produce myelin, called oligodendrocytes, in the central nervous system, CNS, and Schwann cells in the peripheral nervous system, PNS. NGF supports the survival and growth of these glial cells, which are essential for myelination. Moreover, while NGF itself may not directly trigger myelination, it contributes to the overall health and function of neurons. Healthy neurons are crucial for signaling and communication within the nervous system, including the regulation and myelination process. Therefore, it's by supporting neuronal health and function, NGF indirectly contributes to the proper function and maintenance of myelinated nerve fibers. Nerve growth factor is a protein crucial for the growth and maintenance of survival of nerve cells or neurons in the central nervous system. Understanding the detailed pathophysiology of NGF involves exploring its synthesis, release, and actions from astrocytes, the key supporting cells within the CNS. Astrocytes are a type of glia cell that play multifaceted roles in the CNS, including regulating the extracellular environment, providing metabolic support to neurons, and participating in the response to injury or disease. In recent studies, it has been observed that astrocytes are involved in the production and secretion of NGF within the CNS. The synthesis of NGF in astrocytes involves a complex interplay of molecular events within these cells. When stimulated by various factors such as certain compounds found in lion's mane mushrooms, like erinacines, astrocytes respond by initiating a cascade of intracellular signaling pathways. These pathways ultimately lead to the activation of gene responsible for NGF synthesis. Upon activation, astrocytes undergo a change in gene expression, particularly upregulating the transcription of the NGF gene. This process involves various transcription factors, including cAMP. That's CAMP, response-eliminating binding protein, CREB, CREB, and others, which bind to specific regions of the NGF gene, promoting the transcription into messenger RNA, mRNA. Once the NGF mRNA is produced, it undergoes translation in the cytoplasm, where ribosomes synthesize NGF protein molecules. These NGF molecules are then transported within the astrocyte, packaged into vesicles, and subsequently released into the extracellular space. The released NGF can exert its effects through the autocrine and the paracrine signaling. Autocrine signaling occurs when NGF binds to the receptors present on the surface of the same astrocyte that released it, potentially modulating further NGF production or influencing other cellular functions within the astrocyte. Paracrine signaling is when NGF acts on neighboring cells, particularly neurons. NGF binds specific receptors called tropomyosin receptor kinase, or TRKA receptors, present on the surface of neurons. This binding initiates a series of signaling events within the neuron, activating pathways crucial for neuronal survival, growth, and function. Upon binding to NGF to TRKA receptors, several intracellular signaling cascades are activated. These include mitogen activated protein kinase, or MAPK pathway, the phosphoinositide kinase, PI3K, AKT pathway, and others. Activation of these pathways lead to the regulation of gene expression, the promotion of neuronal health, the inhibition of cell death pathways, and enhancement of synaptic plasticity. Furthermore, NGF-TRKA, tropomyosin receptor kinase A receptor signaling, can have long-term effect on neuronal function by influencing gene expression in the nucleus. This includes regulation of genes involved in neuronal survival, synaptic connectivity, and neurotransmitter synthesis. The overall impact of NGF from astrocytes on the CNS is profound. It supports neuronal health, aids in neuronal survival and growth, contributes to synaptic plasticity, that's the ability of synapses to strengthen or weaken over time, and plays a role in the response to injury or neurodegenerative conditions. Understanding the intricate mechanisms of NGF synthesis and action from astrocytes provides insight into potential therapeutic approaches for neurological disorders. However, it's important to note that with this understanding is based in current research. Further studies are needed to fully elucidate the complexities of NGF signaling and its implications for CNS health. The specific formulas for erinacine compounds found in lion's mane mushrooms, Hericium Erinaceus, can vary based on the specific erinacine type, as there are multiple erinacines identified. Gesundheit, such as erinacine A, B, C, etc. Unfortunately, due to its complexity and variability of these compounds, their precise structure and molecular formula aren't readily available in the scientific literature or database accessible up until the last update of January 2022. Erinacines are believed to be bioactive compounds responsible for some of the neurological benefits associated with lion's mane. These compounds have gained attention for the potential to stimulate nerve growth factor, NGF. The mitogen-activated protein kinase, MAPK signaling cascade plays a significant role in transmitting signals from the surface of the cell to the nucleus, ultimately regulating various cellular processes where nerve growth factor NGF binds to its receptor, tropomyosin receptor kinase A, TRKA. It activates several intracellular signaling pathways, including the MAPK pathway. So once NGF binds to TRKA, it leads to an activation of the MAPK cascade, which involves a series of phosphorylation events among various proteins. The MAPK pathway generally comprises of three main kinases: extracellular signal regulated kinase ERK, C-Jun-C, and terminal kinase JNK, and P38 MAPK. Each kinase pathway has distinct functions and downstream effects. ERK pathway, extracellular signal regulated kinase, ERK. Activation of TRKA by NGF leads to the recruitment and activation of the adaptor protein SHC, or SRC, homology and collagen. CHC activates the guanine nucleotide exchange for factor son of 7-less, SOS, which in turn activates RAS, a small GTPase protein. Activated RAS triggers a kinase cascade involving RAF, MEK, MAPK, ERK kinase, and finally ERK. ERK translocates to the nucleus and phosphorylates transcription factors like ELK1 and C-Phos. This leads to the regulation of gene expression involved in cell survival, proliferation, and differentiation. C-Jun N-terminal kinase, JNK pathway. NGF TRKA activation also induces the recruitment of different adaptor proteins such as GRB2, growth factor receptor bound protein 2, and GAB1, GRB2 associated binder 1. These adaptor proteins activate small G proteins, RAS and RAC, leading to the activation of the MAP kinase MAP3K family, including apoptosis signaling regulating kinase 1, ASK1. ASK1 then phosphorylates and activates MKK47, mitogen activated protein kinase 47, which further activates JNK. That's C-Jun N-terminal kinase. JNK regulates various transcription factors such as C-Jun ATF activating transcription factor and others influencing processes like apoptosis, stress response, and cell differentiation. P38 MAPK pathway. Similar to JNK pathway, P38 MAPK activation can be mediated through RAS and RAC GTPase, leading to the activation of MAP3KS. The activation of MAP3KS phosphorylates and activates MAKK36. It's mitogen activated protein kinase 36, which in turn phosphorylates and activates P38 MAPK. P38 MAPK regulates a wide range of cellular responses, including inflammation, stress response, and cell differentiation. The downstream effects of these MAPK pathways triggered by NGF binding to TRKA receptors are extensive and diverse, regulating gene expression, cellular response to stress, survival, proliferation, differentiation, and apoptosis. The specific outcomes depend on the specific cell type, context, and interplay of these pathways and other signaling cascades within the cell.